Welcome, this is Dr. Harries, and today we're going to be talking about Chapter 15. I'm going to have this lecture divided into two parts. In this first part, we're going to discuss mutations. We'll talk about what are mutations, we'll talk about what are some of the different kinds of mutations and what their effects could be. And then in the second half, we'll talk a little bit about how mutations can be tied in with human disease. What are some of the ways to screen for genetic diseases that might be caused by specific mutations? And then lastly, is there anything that we can do to treat those kinds of diseases? All right, so mutations are just defined as any changes in the nucleotide sequence in your DNA. Your genome contains a specific order of bases of nucleotides, and any change in those bases would be considered a mutation. So mutations might have some phenotypic effect, right? Might affect your appearance or the function of your cells or your body in some way, or it might not. It just depends on where that mutation is in your genome. Um, Mutations could be inherited or they might stop with you. So this brings us to our first distinction, which is between what are called somatic mutations and germline mutations. And you might remember we talked about somatic cells versus gametes. So somatic cells are the majority of cells in your body. These are not reproductive cells. So somatic mutations are going to be most of the mutations that you might acquire during your lifetime in those somatic cells, and those are not going to be passed on to any children that you might have. Those mutations would stop with you. Whereas germline mutations is referring to changes in either the gametes, the sperm and eggs, or cells that are producing sperm and eggs. And so mutations there would be passed on to your offspring and are going to be spread throughout the population potentially over time. Either one of these could be a problem. Even somatic mutations, even though they're not being passed on to the next generation, could still cause disease in the person that carries that mutation. So, what causes mutations? If mutations are changes in the DNA, how does that happen? And there's really two ways. So, mutations could be spontaneous, meaning there's no outside factor that's causing that mutation. And really, those would be mutations that are just due to mistakes made by DNA polymerase, which remember is the main enzyme that copies DNA during DNA replication. So even though DNA polymerase is very accurate, sometimes it does mess up. We'll insert an incorrect base, and that would be considered a spontaneous mutation. Or sometimes during cell division, maybe during meiosis, there could be certain mistakes that might happen during the crossing over process, for example, that could lead to mutations as well. And then you have induced mutations. These would be due to some outside agent, typically called a mutagen, and unfortunately there's lots of mutagens, everywhere we go in the environment. UV light is a mutagen. Um, Radiation, both natural and man-made forms like X-rays would be mutagens. There's all kinds of chemicals that can mutate our DNA that we're exposed to on a pretty regular basis. So those would be considered induced mutations. And no matter how hard we try, we're always going to be exposed to induced mutations as we go about our day-to-day. We're exposed to these agents, and we are going to accumulate mutations throughout our lifetime. So, as I mentioned before, mutations can cause disease, serious disease. Things like cancer, for example, is essentially usually a disease that's caused by an accumulation of mutations over time. That's why we tend to see cancer more in older people, because they've lived longer and have been exposed to more mutagens uh, over their lifetime. But if we think kind of big picture in terms of biology and diversity, mutations are actually incredibly important and arguably beneficial. So without mutation, we would have no evolution, right? That mutation is kind of that slow, steady change in genomes that's driving evolution and evolutionary change, increasing diversity over time. Um, and I think I've mentioned this in some previous lectures, but diversity in populations is very important for long-term survival. So even though some of these mutations may be detrimental to the individual that carries them in the short run, in the long term, having mutations and diversity is going to be beneficial. All right, so let's talk about some of the different ways we could group or categorize mutations. And I wanted to start with grouping them based on what the outcome is of that mutation in terms of uh, the protein function in the cell. So really what we're talking about here is mutations that are occurring in genes. And you may remember, what is a gene? Well, it's a portion of DNA that is essentially the instruction manual for building one protein in the cell. So potentially if we change that gene, mutate it in some way, we could also change the protein and its ability to function. So that's the direction we're coming at this from here on this slide. So in the first example, there's what are called silent mutations, and those would be mutations that actually don't change the protein function at all. So here's sort of our control here in the upper left picture, where we've got our DNA being transcribed into a messenger RNA, being translated into a normally functional protein, right? So that's, let's say this is an enzyme and it's catalyzing whatever reaction that enzyme would normally carry out in the cell. Here we have a mutation, but this is a silent mutation because even though we've changed something in the DNA, and that change does get copied into the messenger RNA during transcription, when we went through translation, that change is not really affecting the protein in any functional way. So this enzyme is still perfectly capable of carrying out whatever reaction it needs to catalyze. So if you're gonna accumulate mutations in your genes, I guess ideally you'd like them to be silent mutations, right? They're not affecting your cell's ability to function in any way. The next very common type of mutation would be what's considered loss of function. And what that's referring to is the fact that we're losing the function of the protein coded for by that gene. So here's our mutation in a different part of the gene we see in this image. Again, that gets copied to the messenger RNA during transcription. But now this mutation has changed the protein in some substantial way, and that enzyme has lost some or potentially even all of its function. So we basically broke the gene, broke the protein, right? It's just not gonna work anymore. And then on the flip side of that coin, we could also theoretically have what are considered gain of function mutations. And this would be a mutation that changes the protein in a way that it's actually gaining an extra function or maybe just more of its normal function, sort of becomes hyperactive in some way. Okay. And so really in terms of biology, either loss of function or a gain of function could be a problem. Because really, in most cases, our cells are pretty fine-tuned to work with a specific amount of proteins, certain levels of enzymes. And if we start reducing that through loss of function or increasing that through gain of function, it could be a problem too. I will say gain of functions are going to be much more rare than loss of functions. So I know the sort of silly analogy I like to use is imagine I open up the hood on my car. I'm not an auto mechanic, and I just start changing things around, under the hood. So I'm mutating things chances are I'm probably not going to cause a gain of function and cause my engine to work better and my car to be turbocharged, right? I'm probably going to cause a loss of function and break my car. So it's the same kind of idea in the genome, right? You start changing things, chances are you're probably causing loss of functions rather than gain of functions. But very occasionally, you will get these gain of function mutations. Um, the last group I wanted to mention, and these are even rarer than gain of function mutations, are what are called conditional mutations. And this would be a situation where you've changed the protein, but only under certain conditions, certain environmental conditions. Say maybe the enzyme, the protein functions normally above a certain temperature or below a certain temperature, just as one example. But if you cross that particular temperature threshold, now the protein doesn't work properly anymore. Whereas a normal version of that protein would work properly at on either side of that particular temperature threshold. So that's a possibility as well. So we have those different groups of mutation based on outcome. Now, we could also categorize mutation just based off what type of change, what type of physical change is actually happening to the DNA. And there's really two common groups there, what are called point mutations, where we're just changing usually a single base, a single nucleotide, just one point on the DNA. Um, and that would be in contrast to more dramatic chromosomal mutations where you're changing large segments of DNA. And so I'll show you some examples of both of these. We'll start with the point mutations here. So as I said, point mutation, just a change to a single nucleotide. You know, these could be spontaneous mutations or due to environmental mutagens, as we discussed previously. And really, we're mostly concerned with changes in the genes in what are called the coding regions of the DNA, because that's typically what would actually affect your biology, affect your protein function. That's not to say mutations outside of genes couldn't be problematic, but we know mutations in genes definitely can be an issue. Now, point mutations, there could be sort of different flavors of those as well. So point mutations could be silent, just as we talked about before. This would be a change to a single base, as you see down here in the picture on the left side of the screen. We've changed this one base in the gene marked in yellow here. That gets carried to the messenger RNA through transcription. But it didn't actually change the codon here at all, or it didn't change the amino acid coded for by this particular codon. So our protein is the same as it was originally. So it's completely silent. Protein is totally normal and fine and functional. Then you could have what are called missense point mutations, and this would be a mutation that does change one amino acid in that protein. So here you can see we've changed this codon in the gene and in the messenger RNA, and now that codon actually codes for a different amino acid. So in this particular example, GUU codes for valine instead of the original codon, which coded for asparagine, ASP. So we have that one amino acid change at this one position in the polypeptide in the protein. So that by definition is a missense. We've changed that one amino acid. Now you might ask, okay, well, what's the functional effect of that? And the short answer is, well, it just depends on how important that particular amino acid is for that particular protein. So this could still technically be a silent mutation in terms of the function. Maybe the protein works just fine with a valine in that position instead of asparagine. Maybe it's a loss of function mutation. Maybe it's a gain of function. It really all just depends on the particular example that we're talking about. But there are some famous examples of missense mutations that can cause essentially loss of function and cause human disease. So, for example, the kind of the classic example of a missense mutation causing disease in humans is sickle cell anemia, which is a missense mutation in the hemoglobin gene, beta globin in particular. So you probably remember hemoglobin is the protein that makes up your red blood cells. Your red blood cells should have this nice round shape with the dimple in the middle, and they're carrying oxygen through your bloodstream to all your cells. If you change just one particular amino acid in out of 146 in that beta globin protein, you end up with red blood cells that look like this instead. So they've got that characteristic sickle shape, hence the name of the disease. And so this causes an anemia because these sickle-shaped cells are not as good at carrying oxygen, but it also causes other life-threatening circulatory issues. So these sickle-shaped red blood cells tend to get clogged in little capillaries, can cause, as I said, serious circulatory issues, pain, and it can be life-threatening in many cases, requiring regular hospitalizations typically. So it's definitely a serious disease. Actually, before I go on, I did also want to mention it's kind of interesting that there's a higher rate of sickle cell anemia in African American populations. And that's because there's actually been some evolutionary selection for sickle cell anemia in populations that live in, for example, equatorial Africa, where malaria is a problem. So the malaria parasite, part of its life cycle goes through red blood cells, and it turns out the malaria parasite does not like the sickle-shaped red blood cells. So if you have sickle cell anemia, if you're heterozygous for this mutation, you are basically resistant to malaria. And so if you're living in a part of the world like equatorial Africa where was traditionally killing, lots and lots of people, there was actually some survival advantage to uh, being sick with sickle cell anemia versus being killed with malaria. All right, so we had point mutations that could be silent, no effect on the protein at all. Point mutations that could be considered missense mutations, meaning they change one amino acid in the protein. And then we've got two more categories to mention here. We've got what are called nonsense mutations. And this would be a point mutation that changes a codon that normally codes for an amino acid into a stop codon. So you're basically getting an early stop to the protein. Um, this is almost always going to cause a loss of function in that protein, right? Because you're just not making the full length polypeptide, so the protein is probably not going to work right at all. Similarly, in what are called frame shift point mutations, this is sort of an interesting example where in a frame shift you are either adding a single base or removing a single base from the gene. So in this example here, we've added this extra thymine here. So instead of CCG, we have CCTG now. So this extra base gets inserted. And this is called a frame shift because when we start reading these codons, right, 3 nucleotides at a time, we're going to shift the entire reading frame after that insertion or deletion. So if we look at this, our first codon is fine, right, unchanged. Our second codon is still normal. But now, all of a sudden, our third codon has this extra base inserted, so it's different. And then the codon after that, and after that, they're all going to be different from that point onward. So this is going to cause a lot of amino acids in that protein to be changed, and it is probably also going to result in a loss of function in that particular case. All right, so we have those different types of point mutations. Then, as I mentioned before, we could have more dramatic chromosomal mutations. And so I'll show you some examples of these. These could include deletions, where a whole chunk of a chromosome is lost. In this case, I don't know, imagine these letters are representing, let's say, different genes on the chromosome. So in this case, we totally lost two genes, right? That could be uh, pretty catastrophic depending on what proteins those genes are coding for. You could have what are called duplications, where a portion of the chromosome gets duplicated. So now you've got extra copies of the gene that may be causing a gain of function, right? You basically would be making too much of the proteins coded for by those particular genes. You could have an inversion, where you're not actually gaining or losing any genetic material, but you're just changing its position. So we're taking this little chunk of the chromosome here and flipping it around. Um, and this can still cause issues because the position that a gene is in on a chromosome has been shown to be really important in terms of how it gets expressed, basically how much of the protein encoded for by that gene is produced. So just simply moving gene E from here over to here, as we're doing in this inversion, could dramatically change how much of protein E we're going to make in that cell. So that could be an issue. And then I think the last example I've got here is what are called translocation, which is where a portion of one chromosome, in this case the tail end of the blue chromosome here, CDFG, breaks off and ends up attached to another chromosome. And so again, we're not really gaining or losing genetic information, but we're moving it to another location, similar to what I talked about with the inversion, and we're moving it in an even more dramatic way. So you can have similar problems arise there. So how do you get these big changes? Well, typically these are going to be due to mistakes during the crossing over process that might happen during prophase 1 of meiosis, right, where your homologous chromosomes pair up and we get some crossovers that occur if the chromosomes don't line up perfectly. Sometimes you can get some of these mistakes happening, or exposure to like high energy radiation in some cases can cause breaks in your DNA. And then in the process of the cell trying to fix those breaks, we could end up in some of these situations. All right, I'm going to stop there and we'll pick this up in part 2.